computational science. Please stay here. We're going to invite Fabio will come as well. Fantastic to hear about the future, and wonderful. Couple of questions, and then we'll open it up for questions from for the room, so we have time to also socialize after this event. Question for both of you. Let's start with Elena and then Fabio. Research takes a long time to make an impact. 5 years, 10 years in biotechnology. It's not like we know something today that it's going to change tomorrow. So fast forward 5 or 10 years in the future, what do you see as the potential impact of your research or your team's research, what you're trying to do?
Yeah, sure. So I definitely think that many, so there are a few startups, as I showed, that are directly, well, that have a business model built around the computational platform that they have for like spinning out new products in the market every year, every 2 years, or just, you know, like serially throughout like a very solid computational platform. So I think that the aspect of like having a platform really helps in maybe lowering the time to market, and it's a concept that I know, that I've been hearing that kind of can, decrease this time that you were mentioning.
Yeah, I think it's fantastic because science is becoming so complex and complicated that the human mind, we're reaching the limits of what the human mind can analyze and comprehend, and so we need powerful computers, powerful algorithms, and smart people such as Elena who can actually guide and understand what we're finding in terms of plant biology, gene, and apply it. Fabio, what about you?
So, yeah, among molecular biologists, I think, well, it's said that aging is seen more as a disease nowadays, because when you age, a lot of cells are not functioning properly anymore. And that's also because a lot of disease arise only because you age as a person. And I really think that the CRISPR-like technology will be a way to delay the aging process. This has already been achieved in the eye, in which they're able to make essentially the eye young again. It's not working.
Yeah.
Is able to make the eye young again by essentially activating those genes which are like active during development, and thereby essentially reducing the age from the cells and making the whole eye as an organ younger again. So this has already been achieved by on mouse, making mice younger. And I think maybe in the several decades that this will also be achieved for humans.
Fantastic. So what we're hearing now is the proverbial fountain of youth being created here. We're, I think, stepping on, applying it on first genetic disease where you know there's genetic defect, but then the more complex problems of aging and finding their kind of genetic basis. And so 10 years from now, we might have an ability to reduce the process of aging. That would be really, really fantastic. Back to Elena. Stanford, we hear so much as an iconic place where lots of research is being done, a great place. But what is it like to be on campus? What are some of your experience? What excites you about conducting research at Stanford?
Well, yeah, I would say that there's always something happening on campus, especially from the start of the school year. So for instance, there are always companies or like investors over. We're organizing ourselves in my own department seminar series to bring in more like investors and startups. I'm also part of the Stanford biotech group, which we organize a series of events. So yeah, I love the fact that everybody is looking inwards also about like what's coming out of Stanford, and yeah, that's very nice. Yeah.
Thank you. How about you, Fabio?
Yeah, so I know at Stanford, of course, there's like a lot of money. I think Stanford has combined more money available than all the Dutch universities combined. And the thing I noticed, and also like other lab members that are from Europe, that there's so much money available. So you're essentially able to do like faster science because there's like more money, and just simple things. You're just instead of making it yourself, you just let the company do it, so you can only focus on the important things, doing your whole research. So that's something I really like.
No, you're tapping into something that I personally experienced myself. It was a while ago coming to the United States, having a grant, and suddenly having so many people help you with travel, classes, research, and it really kind of what you're telling me accelerates, and it's kind of a unique aspect of being at Stanford.
Mm-hmm.
Good. And then let's keep it a little bit more on the personal side. You both came from Wageningen. Before that, you were in different places, Italy, for the world. What excites you about living in the Bay Area, and what do you tell your friends at home what it's like to be here?
Well, yeah, for me, still like things happening around everything. Like even though like the Bay Area is like fairly big, right? But there's always something like in San Francisco, in Palo Alto, in Stanford campus. So I really like the fact that one really has to stay focused on their research, otherwise there are like too many distractions, and that's something very nice there. And they have like the Bay dynamic. And it's a lovely place to live. So I am telling this to my friends, and also grabbing as many opportunities that I can from like these months here, and bringing them back to either like Wageningen, my research, or my association. Yeah. Thanks.
Very good. How about you, Fabio? Other than maybe the Bay Area is a little bit more bigger than Wageningen, but
yeah, like you mentioned, there's like a lot of activities. Like you can find people like in every category. You can find them here. Like a lot of excited people, motivated people. It really seems like this really the hub, like worldwide, where all the development is happening. So it's just overall really an exciting place to be. I hope that in the future I come back, of course, to fulfill my dreams.
Excellent, excellent. Well, thank you for that. Maybe I'll turn it over to you if you have particular questions on either what was presented or not presented, any questions you have on genetic research in general, please. Jeanette.
Thank you. I mean, I was going back into classes myself. I was like, ooh, this is so cool. I have 1,000 questions anyway. But to start with you, Elena, we talk about plants. What about soil? Are you also looking into that? Bioengineering, in fact, what soil does to plants, and as a result, plants are very different depending on the minerals and everything that's still in the soil when we grow them, right? That has a lot to do with health eventually. What is your take on that? Thank you.
Thank you so much for the question. I'd say I'm not looking into that specifically for this project, but I completely agree. I know that some, like for instance, actually there are some approaches for like plant breeding, so developing like plant varieties for the future, right, of agriculture, for instance, that are actually not targeting like improving like the plant traits, but like improving the microbiome, like they're around the roots, for instance, and using that as a way to improve nutrient uptake, or like any other like plant coping with different kind of stresses at the root level. So that's definitely an approach, both through like conventional breeding, so just by like natural selection and crossings of plants, but also through genome editing, either improving from the plant side, or like improving from like the microbe side, improving the strains that could go in there. So that's definitely a field of application. Yeah.
Thank you.
Okay, thanks. We'll do our best because of the cord.
I'm back to the 70s with the mics and the lights.
You have some.
Thank you.
Oh, can you?
Yeah.
Yeah. This is a question for Elena. Do you have, with the good scientists, do you have a long-term intellectual property strategy? Because it seems to me you have 1 foot in private industry, 1 foot in academia, and 1 foot in nonprofit. And just to put it bluntly, how do you make sure that these large-scale algorithmic researches are not just sort of taken full-scale by other people?
Can you repeat the last bit?
Yeah, how much are you, maybe I'll put it this way, how much of this is an open source project or more of making things available, making projects available to nonprofits? I just want to know the extent to which you plan to protect your intellectual property.
Sure. Thank you. Thank you. So that's a very good question. So the fact is that we are leveraging initiatives started by universities to put their own patent portfolios to make them available for free to nonprofits. So usually the way that it works is just they have these licenses, which right now go under the umbrella term of like socially responsible licenses. That just mean that if a university has a patent or like a series of patents, they'll just make them available for free to nonprofits to like implement them. So it's not going to be anything about like selling products to nonprofits. We're also not going to have our own IP portfolio, but we want to act as a centralized source of information for like nonprofits to find what could be available for them. For instance, actually, just to make an example, now this is a new trend in Europe, started by the Netherlands in 2019 by taking like socially responsible commitments for like university licensing. But this has been around in the US for a while. Actually, in 2007, a bunch of US universities, including UC Berkeley and Stanford, committed to giving patents for free to nonprofits. But even though like this initiative has been running, we don't know of like a lot of like nonprofits that have been able to benefit from that. So what we just believe is that by pulling all of this knowledge of like what's available in a single place, nonprofits could, take advantage of this because it's already available. Yeah. So technical difficulties are very low. Yeah. Okay. Sure.
So Fabio, is this something also that you get involved in terms of glaucoma research and macular degeneration research with your findings and patenting them through Stanford?
So I'm not entirely certain about the whole procedure, but I know, for example, with the one I'm developing for glaucoma and like age-related macular degeneration, that if it is successful, there's already like a company lined up next year that's able to buy over the IP. So so in most of these cases, when you develop such a medicine, you get like an IP for the certain medicine, and probably then sell it over to a company with really able to scale the whole thing up for like clinical trials and stuff. Yeah.
Good. Thank you. Other questions? Sir?
Thank you. Yeah, this question for Elena. Yeah, I used to work for UCLA School of Medicine and Medical Center, but I'm a data scientist, so I'm very interested in your research. What kind of AI and machine learning technique you use in your research?
Thank you. Yeah. Thank you. So I'd say we want to leverage the knowledge that we have from like existing gene clusters, which are fairly a few. So far I'm using fairly simple machine learning models such as random forest. And currently the current pipeline that we have for genome mining actually does not use any machine learning, so the one that my lab is developing overall. So let's say what I really like about my project is the fact that first the objective is to dig into the biological knowledge and pulling the data together and understanding how to exploit it and develop like what could be like the good like statistical viewpoint around like certain genomic knowledge, but I've just started exploring the machine learning part. Yeah.
Thank you.
Maybe for Fabio, I think I understood also from your talk that you do a lot of data mining and predictive algorithms. How do you apply it in your research? How do you see that?
So currently with my research, I'm not applying it. But I did a lot of like machine learning stuff in the past, and I really see that not only in my field but in many other biological fields, it's all moving to the computer at this point. A lot of like artificial intelligence, like mathematics, like modeling of the biological systems. That will probably be the future. Like from you call it like the wet lab, like all the things you actually do in the laboratory, to dry lab, which will be like all the computer-based stuff. And it's getting more and more dominant. So yeah, also in my specific research, that will probably really be the future. Yeah.
Thank you. I think it's a trend we're seeing in research. Maybe in the 80s and the 90s, the chemists were really important. 2000, 2010, the biologists became very important. And now we see more and more mathematicians being invited into research departments to really make sense of this powerful data modeling, because they become almost a rate limiting step to making the next level of discovery. So it's fascinating to hear that from both of you. One more question? Yeah? Please.
Thank you. Really fascinating, both on the biological and botanical side. I was going to also ask about kind of machine learning, and are there any like open source models like AlphaFold or things that are interesting for your work? And also, now a lot of machine learning and investment is going into generative AI, stuff like DALL-E. And is there any work in generative, like generating different chemicals or pharmaceuticals or different proteins that might be interesting?
Yeah, sure. Thank you so much.
Last question.
Thank you so much. So as I said, I just started digging into the more like prediction part, but I can say that part of like what the pipeline, the pipeline input is something that comes from another prediction software that was developed from my lab, which is called the 2P2 enzyme 2 prediction software, and that kind of predicts what the specific gene do and sort of builds up like the part of the pie, the blue part of the pie that I was showing for plants specifically. And I know that part of in the lab I was like marginally, like I was slightly involved in that part of the project, was exploring the relationship of or just like the validation of those predictions with like protein structure. So like more like relating that with AlphaFold or more like actually Rosetta. So yeah, but that does not directly go into my project. I'd say we're a little bit more downstream. Yeah.
Yeah, just one comment is that indeed, like AlphaFold is really used a lot now, like in molecular biology and also in my department, because it used to be you look at the DNA and might give some like conclusions on a disease or phenotype, and now and sometimes it's just not really that clear. And now with proteins, for example, if they're like misfolded, you can already say something about a disease that is going to develop like in the future, for example, or it might be misfolded, and that might be the cause for the disease, which is just not that apparent in the DNA. And AlphaFold is really helping a lot to in that regard, to really understand how the whole proteins and fit together. Yeah.
One ultimate question.
Yes.
Sorry, by the way, for this back to the 70s microphone. No, I'm a simple economist. Both of you blew my mind with what you were saying, and I'm trying to make it very practical. So I had a question for Fabio. If I come to you in 10 years' time for my rejuvenation therapy, what form would that take, or what form does the eye therapy that you're working on? What is it? Is it a pill? Is it an operation? Is it lasers? What is it?
Like the factor itself that contains the CRISPR is essentially a modified virus, because viruses have evolved to really specifically only target a certain tissue. For example, coronavirus targets the lungs, and a lot of other viruses that target, for example, the heart, the intestine, or the brain. And essentially what you do is remove the bad part, like the actual part that causes disease. You remove that and put the CRISPR system in. So then you have, for example, a virus that only targets the eye, and then you just inject it. That's it.
Okay.
It's just like an injection, like a vaccine, or in the eye, it will be in the eye, but like on any other part from the blood, it will just be like via the blood. So in that sense, it's quite simple.
See you in 10 years.
Thank you, Dirk, for asking the question that's on everybody's mind, but that needed to be asked. So.
And it's amazing because that's actually in the news that viruses are going to really, they're so predominantly around us, and that's like a big cause of disease and mortality. So you're young and you are our hope here, and not for just older people, but apparently also for anyone, right? That's what we're looking at.
So that's also the easiest ways to cure or prevent disease is just rejuvenating yourself, and then it's the easiest way to prevent the disease. So that's what we see like on a molecular level. If the disease is being caused because the cell's aging, just rejuvenate the cell, and then the disease is being prevented. But
plenty of opportunity to talk at the break.
What do you think?
I like to really thank both Elena and Fabio for a wonderful talk. Just come here all the way from Stanford and present to us can be quite fighting. So please give them a big hand of applause. Enjoy with your friends at home, and thank you again on behalf of NUF North California for this great talk. And before we go to the bar, if you want to take your seat, before we done, I just want to introduce very quickly to us the chairman of the NUF, chairwoman of the NUF North Cal chapter. I'd like to introduce to Mrs. Puisan Tan, who will share with you a couple of words about the NUF of North California. Here you go, Puisan.
Thank you, Vlad. Good evening, goedenavond. My name is Puisan Tan, and it's so nice to be back at the Consulate here with so many good friends. I am the chair of the Netherlands American Foundation Northern California chapter. As Vlad has already mentioned, the NUF seeks to further strengthen the relationship between our two countries. We do this through exchanges in the arts, the sciences, education, business, and public affairs. The future of the Dutch-American relationship depends on support for a new generation of academics on both sides of the Atlantic. So we invite you to join the NUF. In doing so, you will undergird a new generation of leadership and educational and cultural exchanges between the Americas and the Netherlands. So let me explain briefly how the NUF is doing that. So the NUF is pleased to provide financial support to researchers such as Elena and Fabio, courtesy of the Dutch to USA internship program. And actually, Elena and Fabio are among the 29 students that the NUF is currently serving. We also have the USA to Holland internship program, and recently the NUF has selected 4 awardees for this. And this program really enables high-achieving, low-income American students, college students, to intern in the Netherlands. So these are only 2 of the grant programs that the NUF runs, but every year the NUF awards over 60 grants to students and artists. So we hope you have enjoyed tonight's program. I just want to say a few special thanks to our NUF NorCal board members, especially Vlad Hogenhuis and Sabrina Perel, who both worked so hard for this event. Thank you, Elena and Fabio. And last but not least, thank you, Consul General Dirk Janssen and the Consulate, its team, and Holland in the Valley for hosting us this evening and for providing the refreshments. And we look forward to your supporting the NUF by becoming members and by attending our future events. So if you have any questions, please come talk to me afterwards, or any of our NUF NorCal board members. There's Jared, Sabrina, Vlad, and Willem in the back. And we also have a table in the back. So the only last announcement is our next event is going to be a beach cleanup at Ocean Beach on December 3rd. So we hope to see you there. Thank you.
And now for the refreshments.
Yes.